Hello, everyone. Happy New Year. So, we are ready to start our new series, which is going to be on genetics. And so, our goal here is to mimic a course that is very similar to an introductory level genetics course in a biology department in undergraduate universities. And so, it's the closest equivalent to Genetics 101. So, here we go. So the textbooks that I'm going to be using are both available online, free. These are open textbooks, online open textbooks. I will have the links for these down below in the description so you can access them easily. So there are two different ones that we're going to be using. One of them is this first one, the Nickele and Barrette online genetics, and I will show you. This one is very simple, easy to understand. I love the way it is written in a very kind of clear form. Each section is very small, easily understood, and you can pretty much like follow along with it. And we're going to be using a lot of this book in the beginning chapters or in the beginning lectures that I will be posting. I'll have more of a mix of the two later on as we get more detailed into, more into the meat of genetics. But in the beginning, because of its simplicity and ease of understanding, I'm going to rely more on this particular book. So here's an example from what we're going to be working on today, which is kind of an overview introduction to DNA and genes. Um, and so if we click on any one of these sections, you will see it almost reads like a very good notes from one of the big textbooks. So it has important terms, their meaning, and enough information to give you a basic understanding of the topic. And then again, like I said, for more detailed understanding, we will be going to a different textbook along with the information that I will provide you through chapters from other sources or primary literature articles that I might post. So the second book that I'm going to be using is this Open Genetics. This is Opentexts Genetics textbook that is collected and edited by Michelle Markstein. Um, this one is a lot more detailed. It goes through each topic in a lot with a lot more description, a lot more kind of background information. And so this is the one that we're going to be working with more as we go further into genetics and as we begin to understand the different topics within it. And we begin to work with actual problem sets and actual theories behind different topics. So that's the one that we're going to be working with a little bit later in the semester. So in the beginning, we're going to be focusing more on this first book by Nickele and Barrette. Um, so that's the textbooks that we are going to be working with. Again, the links will be down below. So this week, we are going to be pretty much just reviewing what we already know in many cases and putting it in the context of genetics. So today we are going to be just kind of taking an overview of some of the basic terms. We're going to be focusing on DNA and genes. So we're going to talk about DNA as the genetic material. We're going to review structure of DNA, talk about how genes were discovered as the basic unit of inheritance and what their functions might be inside our bodies. And then towards the end, we are going to be looking at specifically nuclear genome, what is there, and then we're going to be ending with some model organisms that are um, important specifically in genetic research and all types of research, but we're going to be looking at them in a little bit different light this time, focusing more on what type of um, needs each one of them cover as far as genetic research is concerned. So starting out, what is genetics exactly? So genetics is basically a scientific study of heredity and our inherited characteristics. It doesn't just include learning about the DNA or learning about how we look and how we are different from other organisms and other species, but also looking internally, again, at a more molecular level, looking at individual genes and individual portions of our genome, the DNA, and understanding their structure and function and how different parts of our genome interact with each other and work together to make us who we are. We also want to, as part of genetics, we like to understand how our genomes change over a small period of time or over millions of years or hundreds of years, to adapt and change with the environmental changes that they are exposed to, and how that affects the generations and the species in a short and long-term manner. Now, heredity specifically, which is a big topic in genetics, is basically how we pass our physical and mental characteristics through the genes, through our DNA, through our genome, from one generation to another within a single organism. This knowledge of heredity and this knowledge of how the genomes are, how the genes in our DNA are affecting these physical and mental traits allow us to manipulate at times some certain aspects of it and select from naturally existing phenotypes or naturally existing combinations of physical and mental characteristics to choose for those that are more favorable to us as an individual or as a species, as needed. However, now as we are learning more and more about DNA and are able to understand how it works more and more with increasing research, we can also change this ourselves. Not only can we manipulate and select from naturally existing combinations, but we can create our own new combinations that we may find better or that we may find more advantageous to serve out the goals that we want to be working towards. So, obviously, that opens up a lot of ethical issues as well, that we need to be mindful of, and it is very important that we keep that in mind moving forward as our capabilities increase, as our knowledge is increased, our um, capabilities are increased, we have more tools in our belt, that we are using this new knowledge wisely and ethically and only for the betterment of the society and the world at large. Okay, so moving along. The first thing that we're going to be looking at is how did we identify DNA as the genetic material to begin with? So it was not always known that DNA is the main genetic material, right? Initially, there were many other theories about, or many other hypotheses rather, about what could possibly make us who we are, whether it was an essence of some sort or something given by God, that was not in the form of genes or DNA. Uh, Mendel postulated first the existence of genes, but did not know what they were made of, where they came from, whether they were proteins or DNA or lipids or anything else. Others also studied this at the same time. Mesher studied nuclein from pus, and that was found in surgical bandages, and initially thought it was DNA, but then he changed his mind and was like, no, that's too simplistic. So that wasn't possible. So between 1910 and 1940s, the bigger thought was that proteins were the genetic material. mostly because proteins were a lot more complex because they were made up of these 20 amino acids, where DNA was only made up of 4 nucleotides. And so nobody thought that something as simple as that would make up for somebody as complex as the organisms that are present on Earth. So the first of these experiments that started to look at genetics or these genes was the Griffiths transformation experiment from 1928-29. In their experiment, they were working with Streptococcus pneumoniae, and they had seen two different types of Streptococcus, some that had rough edges on the plate when they were cultured, and others that had smooth uh, look. The colonies were very smooth looking. And they found out that um, the colonies that were rough edges, if they were used to infect a mouse, while they may have gotten sick, but they were fine, they were able to recover from that infection. However, the smooth colonies, on the other hand, caused a fatal disease and fatal infection in the mice, and they died. So they would inject the bacteria from these colonies, either just the rough bacteria or the smooth bacteria, into these mice and look at what happened. Now, next, what they did was they went ahead and they heat treated the smooth fatal strains of bacteria before they injected them into the mouse, which would have obviously killed the bacteria itself. And of course, mouse was fine, no infection, everything was good. They then did something more interesting. They mixed the two types of bacteria, the rough and the smooth, um, and they made sure that their smooth bacteria were heat treated first. So now those smooth bacteria were technically dead, and they have added the rough bacteria to the mixture that are still alive. And these mixed cultures were then injected into the mouse. Well, lo and behold, what they found was that the mice died. And when they looked at the culture inside the mouse, when they took the culture from the blood of those mice, they found that those mice actually had smooth bacteria inside them. So somehow the heat treated S strain, whatever was left behind, something non-living inside it at that time, obviously, was able to transform the rough bacteria into the more virulent, more fatal strain that then caused this infection in the mice and killed them. So obviously, that was extremely interesting for multiple reasons, right? But specifically here, that means that there is some non-living component in the S strain that can be transferred into the living R-type bacteria and transform it into the more fatal S strain. So that's as far as they went with their experiment. And then it was several years, over a decade actually, before Avery, McLeod, and McCarty specifically showed that DNA itself was the genetic material in 1944. What they did were they took those same S strain virulent cells and they completely broke it apart into its various parts. They fractionated them into different classes of macromolecules. So you know the different classes of macromolecules that are present, and those have, your lipids, your carbohydrates, your proteins. And so they had the proteins, the lipids, and the carbs separated out. And they also separated out the DNA. So the nucleic acids had both the DNA and RNA, and they took out those two components separately. And then they took each one of these fractions, and they mixed them with our strain of bacteria to see which one of these fractions were able to transform the R strain into the smooth fatal strain of bacteria. And they found that only the DNA was able to convert or transform the rough bacteria into the smooth virulent strain. They then took the cell extracts that they had of the S-type cells and they treated them with DNase, which is an enzyme specifically designed to digest the DNA. So at this point, there will be no DNA there at all. And they checked it again. And this time, they were unable to transform the bacteria into R strain, from R strain to the more fatal smooth strain. And this confirmed essentially that DNA was the transforming factor. That's the genetic material and not the protein or RNA or any of the other components that they were looking at. Because the only time that they were able to have smooth bacteria form or the R strain transform into the S strain was when there was DNA specifically present from the smooth cells that could be picked up by the R strain to transform it into the virulent strain. So the conclusion that they drew was the molecule that carries the transforming material is the DNA itself. So a few years later, Hershey and Chase showed conclusively that DNA was in fact where genes were formed, where genes were found in the system. So in their experiment, what they looked at was, again, they were working with bacterial cultures, but this time they're working with E. coli, and they were working with a particular type of virus, a bacteriophage, so a virus that will specifically infect bacterial cultures. And this was the T2DNA bacteriophage. So it's a, a virus that contains DNA inside it. It's a DNA virus. And they, first of all, grew the DNA, they first of all took the DNA in the bacteriophage and they cultured it. So the DNA bacteriophage viral cultures were made either in the presence of radioactive isotopes that were P32 or phosphorus, which would specifically mark the DNA because of the backbone that it has, the sugar phosphate backbone, or they used S35, a radioisotope, which would specifically mark proteins because the DNA does not contain any sulfur. And so once they had labeled the bacteriophages, they then infected E. coli with these cultures, so with these bacteriophages. So they were either the P32 labeled viruses or the S35 labeled viruses. And they were allowed to infect E. coli. Once they had infected the E. coli, and here you can see an electromicrograph where you can see the bacteria and bacterium, and then you can see all the viruses that are bound to it. The viral heads are visible in this. So these viral heads were then sheared off the bacteria so that there was no body of the virus left behind on the bacteria. Only the DNA or only whatever went inside the bacteria to infect the bacterium is present at this time and nothing else. So here is a cartoon of what it would have looked like. You had your bacteriophage, you had whatever transforming material was in there, and it would have been bound to the cell wall. The virus only takes the transforming material into the cell. The rest of the body remains outside, which is then sheared off. And next, these bacteria that were infected were pelleted down and they were examined to see what was left behind. And what they found was that only thing they found inside the bacteria were P32 and no S35, which meant that DNA was the transforming material that went inside the bacteria and not any of the protein associated with the virus itself. So all the viral body was made up of these proteins, the protein coat, the sheath, everything was protein, but the inside contained DNA, which is what was injected into the bacteriophage. So this conclusively shows that the DNA is, again, the transforming material, and that's where the genes are going to be hanging out. So once those were done, they then started to look at what exactly DNA does once it is there inside a cell, and how is it getting replicated so that it can be transferred to the progeny or to a different place. So there were three main models or three hypotheses that people had about how DNA might replicate in order to be passed to the next generation. The first one was a semi-conservative model where it proposed that the two strands of the DNA molecule would separate during replication, and then each one will serve as a template to create a new complementary strand. The second model they had was a conservative model, which proposed that the entire strand is just act as a single template and an entirely new strand is formed as a result of replication. And the last model they had was a dispersive model, where they said that the two strands kind of randomly have different sections that act as template and other sections that act as replication area, and they're just kind of replicated and reassembled with the new duplexes, kind of a mix and match of old and new on each strand. So to examine which one of these three hypotheses was correct, they used a very kind of ingenious method where they had some bacteria that was grown either in N14, just normal, nitrogen containing medium, or they had N15, a radioactive version of the nitrogen present um, in the soup, in the culture, and so that would essentially make the bacteria incorporate either this normal or radioactive version inside the DNA. And so then when they isolate or when they use a cesium chloride density gradient, they can look at the bands and they can see where it is. So if it was, in fact, a conservative model where you have an old and a new, all the old bacteria, because they were non-radioactive, would give one band, while the heavier new bacteria would all have a higher band with the N15. If you had the dispersive model, you would just get a single band, which is neither at N14 nor at N15. And if you had a semi-conservative one, you would also get a different banding pattern. So they used this way to look at, they first just did kind of a check to see that system works. So if the bacteria are grown in N14, you have lower density and it's closer to the top of the tube. If you use the N15 heavier DNA, you have the band that is higher density, so it's going to be further, it will sink more into the tube, and you will have a lower band. So you can definitely separate the two out. So this experiment was done by Messelson and Stahl, and they were basically running this experiment to check these three hypotheses. And with their experiment, what they were able to do was they were able to definitely rule out the conservative DNA replication model. So here is the experiment in general. So they had bacteria that was grown in the light medium, and they were able to see where the band would be after centrifugal force was put in. And when it was done in heavy medium, then the centrifugal force would push it further down the tube, and they could see where that band would be. So they would grow the bacteria in heavy medium first, and then they would transfer it to the light medium. Now, bacteria are great in the sense that they have a really fast replicative cycle. So within 20 minutes, they're able to replicate their DNA entirely. And so the first thing that they would do is that within 20 minutes, they would have gotten one cycle at that point of replication done. So they looked at it and they found that they got a single band, kind of halfway between where they expected the light versus the heavy band to be. So that meant that it was definitely not a conservative DNA replication. It had to be either dispersive or semi-conservative. And so that is one way that they were able to figure out which one of the models is most likely to be the case. Now they then allowed them to grow more time in the light medium. So they started off with 100% heavy DNA. And within 20 minutes, like I said, they saw a shift and a single band indicating that it was half and half at that point. They then allowed it to go another cycle, so for 40 minutes, and they looked at it again. And this time they saw two bands, half heavy and half light. When they continued this further, they saw further reduction of the heavy, so they only got a quarter heavy, and 75% of the DNA was in the light band, and then so on and so forth. So each generation, the amount of heavy uh, media or heavy DNA reduced by half. This meant the only way that that could have been happening was through semi-conservative replication. Because if it was dispersive, you would not have gotten to this point. You would have seen just a 50-50 ratio all throughout. And that wasn't the case. So um, through these experiments and more like it, they were able to identify that DNA replication is a semi-conservative process. Now, again, this is not a proof of the model. It's just very, very, very strong evidence for it. So hypotheses are never really proven. They can either be disproven or they're supported. And in this case, the data supports a semi-conservative process. We always have that small chance that new information comes into being where we realize that there's some other hypothesis or there is a better explanation for the data as well. So here is an example of what they were looking at. So they had the first set of replication where, this is the original bacteria. It was all heavy. The first set of replication where both contained one heavy and one light, which is why you got a single band. In the second round, you only had two heavy strands left, and then all four had one new strand and one of the strands from the first generation, which were also light. So you would have 50% completely light and 50% at that intermediate level. Nothing is 100% heavy anymore. And then when you continue further down, you see even more reduction of that. So again, only those two initial bands from that first replication that contained one of heavy strand. Everyone else is going to be at that lighter level. So you're going to get a light band and a heavy band and an intermediate band. So this all is very good. Definitely DNA is the genetic material, but there are always exceptions to the rules. RNA and proteins can be genetic material, can act as genetic material as well. The biggest example of this are RNA viruses. So the RNA viruses can be single or double-stranded RNAs, and they are the ones where RNA is the genetic material. That's all they've got. Your examples are SARS, COVID, influenza, Hep C, polio, all of those are retroviruses, um, or are RNA viruses, as well as retroviruses like HIV and AIDS. All of those use the RNA, and that's what's injected into the cell that is getting infected. Once it is infected, there will be DNA sometime in that process, at some stage in their life cycle, where it is then replicating the RNA genome into DNA that is then used as the main template for making more viral particles. Another exception rather, is prion infections, in which you have specifically proteins. No nucleic acid is present. It is a misfolded protein that is causing the infection. The biggest example of those is the bovine spongiform encephalopathy, or BSE, which is also known as mad cow disease in cattle and deer, and CJD in humans. Any prion disease that we look at, they work by altering your brain or neural tissue, and that's how they cause the disease phenotype, and are usually, well, currently at least, they are all untreatable and eventually fatal. Okay. Okay, so before we go any further, let's go through and review the structure of DNA. That's what we're going to be doing next. So let's go ahead. So DNA, building blocks of DNA are basically your nucleotides. A nucleotide consists of a nitrogen-containing base, which are what we're going to be talking about, the main bases that are part of the DNA. And then it has a sugar phosphate backbone that they are going to be attached to. So the nucleotides are the subunits of your nucleic acids, whether it is DNA or RNA. So in DNA, you have 4 bases that are going to be part of the story. You'll have your adenine and guanine, which are the purines. These are the 2-ring structures. And then you have cytosine and thymine, which are your pyrimidines, which are single ring structures. In RNA, you have an additional base called uracil that takes the place of T, but we're not going to be talking about that right now. We're just going to be focusing on the DNA. Now, the other thing that I would like you to review and be mindful of as we move forward is the nomenclature for the different types of nucleotides. When you have the base by itself, it's going to be called by its base name, which is adenine, guanine, cytosine, or thymine, or uracil if it's you. When you have a nucleoside, on the other hand, it is the base that is already attached to its associated sugar molecule, whether it be deoxyribose or ribose. And so it will be called by the nucleoside name. So adenine becomes adenosine, guanine becomes guanosine, cytosine becomes cytidine, and thymine becomes thymidine. So that's, if I ask you for the nucleoside name, you will give me the nucleoside version of the nomenclature, adenosine versus adenine, if I'm just asking you for the 4 bases and their names. They are abbreviated by their first letter, and so it's A, G, C, U, and T. The next thing you want to know is the nucleotide version of the name. A nucleotide is when the base is attached not just to the sugar, but also to the phosphate in the backbone. And that is when you will be calling them nucleotide. The nucleotides and the derivatives are abbreviated again similarly by the capital letters. So the nucleotide associated, for example, with adenine would be adenosine because it's the nucleoside, and then the phosphate, monophosphate if it's just a single phosphate, diphosphate if there are two, triphosphate if there are three. So AMP would be adenosine monophosphate. ATP, something we all know about a lot, which is our basic currency in our cell, the energy packets, is adenosine triphosphate. So be clear on the nomenclature as we move forward so that you can identify when we're talking about the base versus nucleoside or nucleotide. Now, the base pairing that enables DNA replication and allows it to be semi-conservative, as we saw, is dependent upon Chargaff's rule, which essentially Chargaff's rule was that he observed that no matter what piece of DNA he takes apart, there will be equal amounts of adenine and thymine at any given time, and equal amounts of cytosine and guanine in a particular strand that he picked apart. And using Chargaff's rule, Watson and Crick, when they were building the DNA, realized that that meant A and T can pair together and C and G can pair together. In that case, the width of the DNA remains constant all throughout because in each case you have one pyrimidine and one purine coming together. And because of that, each strand of DNA can act as a template for the synthesis of new complementary strand, thus allowing for semi-conservative replication to occur. So the nucleotides are joined with phosphodiester bonds in both DNA and RNA. So you have your phosphate group that is binding to the sugar molecule at the 3' end of one to the 5' end of the other. And this is just an example of how that would happen, leaving the bases free internally to kind of interact with the opposing strand. So the secondary structure of DNA is basically how, once the two strands are built, how are they going to interact with each other and how are they going to be placed inside of, in a stable way, inside the nucleus. And so the early data provided some of these clues. So one of the first thing was that chemists knew that the nucleotides had a particular structure that allowed for interaction with different nucleotides, and that the DNA had a sugar phosphate backbone. Then Chargaff established that the number of purines and pyrimidines is always equal in any given strand of DNA, and also that there are equal numbers of T's and A's and equal number of C's and G's, as we talked about. And finally, Franklin and Wilkins used X-ray crystallography to measure distances between various atoms in a DNA structure and looked at the shape of that X-ray crystallography image to predict that it was most likely a helical structure. Initially, they thought there were 3 strands, and then they started to look at a 2-strand structure, which we now know is what it is. So DNA molecules are typically going to be double helices. And so here is that famous image from the X-ray crystallography done by Franklin Roosevelt. And here you can see this particular type of structure. This is an X-ray diffraction from a hydrated DNA fiber that's being looked at. And looking at that central cross image, it is what allowed them to think about it as a helical structure. And then you can now see all these kind of strong arcs that are forming, which we now know are because of the stacks of DNA, the base pairs that are present inside it. Looking at it in this cartoon image, we see our sugar phosphate backbone that is of the two strands that are kind of wound around each other. And then you have the base pairs inside that are interacting with each other. And they knew from the, again, the X-ray crystallography image, they were able to measure the distance between two of these backbone sugar bases, base structures, and they were able to see this 3.4 angstrom distance spacing between the two sugar molecules, which is maintained in this case. And your bases are at 90 degrees from that sugar molecule. So here it is again, here it is unwound and just kind of parallel. You see the two strands of DNA. The two strands of DNA are kept together through hydrogen bonding between the complementary strands of DNA. And one of the things that was discovered as they were building the structure was that the only way this system would work was if those two strands were anti-parallel to each other. So one of the strands goes from 3' to 5' direction, while the other one has the opposite, starts at the 5' end. And these are then wound around to create the double helix that we know. Looking at it internally, you can look and see how cytosine and guanine are able to interact with each other through 3 hydrogen bonds, because there are 3 hydrogens that are available here in this guanine that can in this cytosine that can interact with the guanine. And similarly, in thymine and adenine pair, there are 2 hydrogens, one in adenine and one in thymine. Here you had 1 hydrogen in the cytosine and 2 in guanine that were making that interaction. So there are 3 hydrogen bonds that are present in CG pair and 2 in AT. This also lets you know that this is a slightly stronger interaction than AT. So DNA, the more CG is present, content is present inside a DNA strand, the stronger it's going to be and the higher melting temperature will be needed to separate those two strands. And that will become important later on when we start talking about different techniques where we need to melt the DNA and then make a copy of it, as in PCR. So the two strands, the double helix that comes out of the DNA, is basically the 2 DNA chains that are coiled around a common axis. Their chains run in opposite directions. So one is 3' to 5', while the other is 5' to 3. So they are basically anti-parallel to each other. The sugar phosphate backbone is what is visible towards the outside, while the bases are stacked on the inside of the helix. The entire helix has a fixed diameter because every time there's one pyrimidine and one purine interacting, keeping that diameter constant. And the way it loops around, the way the double helix is formed, you end up with a major groove, a larger groove, and then a minor groove that is followed by it. So the diameter of your DNA helix is going to be 20 angstrom or 2 nanometer. So just to review the DNA structure, James Watson and Francis Crick were the ones that built that first model of DNA, where they showed the 2 strands held together by hydrogen bonds between pyrimides and purines. The complementary base pairing, which is called Watson and Crick pairing, based on Chargaff's rule, where A and T are paired together and C and G are paired together. The strands are anti-parallel to each other, and the strands form a double helix with the sugar phosphate on the exterior and the nitrogenous bases facing the interior of the structure. So moving from here, we are going to now look at the DNA a little bit closer and see how genes were identified as the basic units of inheritance. So in olden times, before the experiments were performed to examine it any further, there were two different, again, hypotheses around for this, how things were inherited, how characteristics were inherited. One was the blending theory, blending inheritance, and the other was particulate inheritance. In blending inheritance, it was basically thought that some kind of essence is mixed at the time of mating from each parent, and that mixing is a little bit like coloring with paint, mixing paints. So once the blend was formed, once that new blended essence was formed, it will have its own properties, and the individual characteristics of each one of your parents could never be separated again. On the other hand, Gregor Mendel, the monk that performed meticulous, very detailed experiments using peas, pea plants in many cases, um, was able to, had a different idea. And he was able to show that with the different crosses that he performed using his different colored plants and peas, different features, different physical features that he could monitor in pea plants. So he showed that if he took white flowering plants, pea plants, and crossed them with two purple flowered plants, where either a purple flower plant was true breeding, so it only produced purple flowers all the time, which meant that it never had a white parent, or a purple flowered plant where it started, where it had either one purple and one white parent, then he was able to observe white flowers being produced at some point. So if he took a purple plant and it crossed it with a white flowered plant, the first generation, it would only get all purple plants. However, in subsequent generations, he began to see some white plants come out as well. So it was not that white flowers were irreversibly lost. They could come out again. It was a trait that could be seen again, though maybe not in every generation. And this allowed him to disprove the blending inheritance and instead come out with his own hypothesis, an alternative concept, which was called the particulate inheritance, in which heredity is essentially a product of some discrete traits that are independent and that are controlled in that way, and they can come out in subsequent generations in one progeny or another. So how exactly do these traits form through genes? So building on these initial observations, Beadle and Tatum worked out a new hypothesis. So long time before Beadle and Tatum, in 1908, Ajar described certain types of diseases like albuminuria, which is black urine disease, as an example of how genetic defects can lead to a disease phenotype because of a lack of a particular enzyme or necessary product in our bodies. In 1941, Beadle and Tatum built on these connections through their experiments using a very simple model system in the form of a fungus, Neurospora crassa. They hypothesized that each enzyme that they look at will act in a biochemical pathway um, and will be encoded by a different gene. So no enzyme is going to be coded by, no two enzymes will be coded by the same gene, and each gene will only code for one single enzyme. And so to examine this hypothesis, they used this mold, Neurospora crassa, which and they used a particular strain which is normally prototrophic. That is, this particular type of fungus in general is able to synthesize its own amino acids and grow on minimal medium. So just as long as it's given just a few minerals, some simple sugars, and one vitamin, which is biotin, it can pretty much make its own things, make all the amino acids it needs, and survive on its own. So what they did was that they decided to expose Neurospora to X-rays, with the idea that X-rays will obviously cause random mutations, breaks in the DNA, and potentially these breaks are going to be all over the place, right? So each break is going to be in a different gene potentially. And so if they examine hundreds of these mutants, they should be able to look at different genes within the organism. So what they observed after examining hundreds of these mutant molds is that most were still prototrophic. They still worked out fine. They were still able to live on minimal media and survive. However, some now became what we call oxotrophic, which means they can no longer grow on that minimal media without any of those amino acids that they needed, but instead required complete medium, which was supplemented with nutrients to grow. Now, they also discovered that they could just take some of these mutants and they could rescue them with just adding a single nutrient that is supplied in the minimal media. So, for example, in the case of this particular mutant, once they mutagenize it and they transfer that single spore and incubate, if it was not able to grow on minimal media and could only grow on complete medium, they would then again grow it in a new culture of minimal media with just one of the amino acids provided to them. And they looked at when they could see this mold grow again. And so they tried, for example, all these different amino acids, and it only grew when it was supplemented with the minimal media was supplemented with arginine, which meant that the original mutation that happened in that particular strain or in that particular spore that they were looking at was in the arginine pathway, so that it was no longer able to make its own arginine. It was able to make all the other amino acids. It didn't need them, but it did need arginine to be provided to it because otherwise it wouldn't work. So their hypothesis, which was that one enzyme, one gene, was led to a whole new world of genetic analyses, right? Where now you could dissect biochemical pathways by examining these kind of mutations and by examining various mutants and kind of looking at them one at a time to build the pathways, to build different portions of the pathways. So Serb and Horowitz a few years later tested the ability of amino acids to rescue different types of oxotrophic strains. So starting off again with that arginine mutant that we were looking at earlier, they added one of each of the amino acids to the minimal media, just like we talked about before, and recorded if it was able to restore the growth or not. And if it was, then they knew that that's the pathway that was broken and they would call it by that mutation. So if a mutant only grew in the presence of arginine in the minimal media, then the original mutant was mutated in the arginine pathway that was required to build arginine. Now they extended it one step further and they said, well, let me look at that biochemical pathway and see what other intermediate steps are present. So maybe I can figure out which step is broken. So they knew that to create arginine, there were two intermediate steps, which was the CYD and the ornithine and citrinine. And so they next made new cultures where they just supplemented one of these intermediate steps. And they looked at it again. And this time you had the three mutants, and one of them grew regardless of whether arginine was provided by itself or the set or the ornithine, and they were all fine. Gene B mutants, so mutants in a second mutation in that arginine pathway, couldn't grow when only ornithine was provided, but did grow when said or arginine was provided. So that meant that it was definitely not a mutation in the ornithine gene, gene A, but it was in gene B instead, since they required citrullina to be present before they were able to build arginine by themselves. And then gene C mutants wouldn't grow with either one of the intermediaries because both um, pathways were broken and instead required arginine to be present there because they couldn't make it themselves at all. So based on these results, they could pretty much deduce which mutant had what mutations within that pathway as long as they knew the pathway and the intermediate steps within it. And that was, again, a big advance. So knowing all this is how the field of genetics started, which is what we're going to be looking at this entire semester. And we're going to be focusing a lot of our time on the big genome that is present inside each organism in the nucleus, which is basically the nuclear genome. So we should probably get to know it a little bit more. So eukaryotic DNA is packaged in the nucleus in the form of multiple chromosomes. So if you were a prokaryote, you would just have a circular genome present in that space since you don't have a specific nucleus or membrane-bound nucleus. But eukaryotic DNA is packaged into multiple chromosomes, which are then housed inside the nucleus of the cell. Humans, for example, contain 3.2 times 10 to the 9 nucleotides. That's a lot of nucleotides that are packaged into 24 different chromosomes, 24 pairs of chromosomes. These chromosomes are packaged inside the DNA and the nucleus in the form of chromatin, which is a complex of DNA and proteins combined together to package it in just the right way. We will be talking about that later in the semester as well. And the chromosomes are inherited from our parents, one from our mom, one from our dad, in the form of homologous chromosome pairs, which is what you're seeing in this karyotype shown here that is visualized through chromosome painting. Now, chromosomes are essentially just long strings of genes. So it's a long strand of DNA and it will have multiple genes along those strands. And they have those two arms. Each arm has its own different genes that are present. They can be in either orientation within it. A gene is nothing more than a small segment of DNA that contains instructions for making some type of a protein or RNA molecule. Some RNA are the final products, but majority of the RNA are used to make proteins down the road. They are translated into proteins, and that's going to be your final product that does the work inside the cell. So here, as an example, you have 4 different genes on this particular strand of DNA that make 4 different RNA transcripts. Obviously, 3 of them end up building protein out of their transcript, while for gene C, the RNA is the final form, and that's what's going to be doing the work. So one of the things that we use with the genome is to calculate the C value of our nuclear genome. C value is nothing more than just the simple amount or content of DNA contained within a haploid nucleus, for example, a sperm or an egg, a gamete, or basically 1/2 the amount of a diploid somatic cell in a eukaryotic organism. A typical mammalian cell, for example, contains 6 picograms of DNA in a diploid cell, while a gamete will contain 3 picograms. So the C value of a mammalian cell, in this case of a human, for example, is going to be 3 picogram of DNA. The C value is given either in picograms or in megabase pairs or MBPs, and that's going to be the number that you're looking at. So generally speaking, you would expect that the more complex an organism, the more DNA should be there, right? And so there should be some type of a relationship, hopefully a linear one, between genome size and the complexity of an organism. So basically, the more complex organism, the larger the C value. Smaller organisms like viruses and bacteria, you would expect that they would not have room for extra stuff, so they will have a smaller C value. And generally speaking, that is kind of sort of true. However, there's a lot of ambiguity there, right? So you can see over here, for example, a mouse, a musculus versus a Homo sapien, both have about 25,000 genes, and their DNA content, while it is a bigger DNA, C value for a Homo sapien, the average gene density is similar in both cases. Um, so there are some other ways that we can also look at this nuclear content. One of them is G value and another is I value. So G value is, instead of looking at the DNA content, you're looking at the number of genes specifically found in a haploid genome. And this includes any known genes as well as predicted open reading frames that are present within it. And the I value, which is another measure that you can have, is not just looking at the open reading frames, but also looking at effective number of genes that you can get out of them. So that would include alternative splicing from the same RNA transcript to make multiple types of proteins, post-translational modifications, multi-domain proteins, any type of redundancies that might be present in our genome, as well as things that are regulating our gene expression and gene interaction. So that kind of encompasses a lot more than just simply looking at DNA content or G value. In general, C and G values are not very reliable if you're going to compare them to complexity of the organism. As you can see here, that while generally speaking there is that trend that more complex organisms have higher C values, there's still a lot of variability, and some of these changes cannot be explained simply by differences in number of functional genes. For example, here you have angiosperm plants that have this humongous genome and great variability in the size of a haploid genome in base pair. So the reason for this discrepancy at the end is that especially in higher organisms, the more complex organism, much of your DNA is not just coding for amino acids and proteins, but rather it has other functions, whether they are regulatory functions or post-translational modifications or just controlling the genome overall. They have other functions, and that's why they are there. In addition, much of your non-gene DNA is actually transposable elements of various types that have come into our genome as we've evolved over time. And they are interesting class of self-replicating DNA elements, but they are not necessarily part of our everyday conversation when it comes to functional genes in our body. You also have other highly repetitive sequences that are present that are also part sometimes of regulation, sometimes cause of disease, who knows. A lot of these other elements that are there that can also be contributing to these numbers and the variability that's present within it. And so we will talk about those transposable elements definitely later on in the semester as well. Now, eukaryotic genomes don't just contain nuclear genome. They actually also contain non-nuclear genomes in the form of mitochondria and chloroplast. So mitochondria and chloroplast each have their own genomes, and these are very similar to our prokaryotic genomes in that they're circular and they're relatively small, but they contain important genes that are important for our survival, and mutations in these can also lead to disease, sometimes fatal diseases. These are originating again many times. The endosymbiont hypothesis, which is the most prevalent hypothesis about them, states that they at some point were prokaryotes. They originated from prokaryotes and were incorporated into eukaryotes through engulfing and ingesting. These mitochondria and chloroplast pretty much replicate very similar to binary fission, like you would see in prokaryotes of many types. And they can replicate on their own inside the cell. And one cool thing about these is that they are often inherited uniparentally, which means that you don't get them half and half from mom and dad like you do with your nuclear genome, but rather only from male or female parent. In humans, for example, the mitochondrial DNA will come just from the mom and not from their dad. Um, so that is an interesting tidbit about these non-nuclear genomes. Chloroplasts are only found in plants, or photosynthetic plants, so they would have similar idea as well. The last thing that we're going to be talking about today is the different model organisms that people use, geneticists use specifically, and why they choose to use these organisms. So the most commonly used model organisms, there are quite a few of them actually at this point. Um, Most of them are chosen because of their simplicity or their ease of use. The simplest one that we use is a prokaryote, E. coli, and it's the most simple genetic model organism. It's often used for cloning purposes to take DNA sequences from one model species and to clone it into the E. coli and then use it for other purposes. Another one that is, again, a very simple model organism is a Saccharomyces cerevisiae, which is a yeast. It is a good general model system for basic functions of eukaryotic cells because many of the biochemical pathways are maintained in yeast that are also present in higher organisms. We also, geneticists also use C. elegans a lot, which is a round form, which is a great model system to study the development of multicellular organisms um, because it is the entire life cycle of C. elegans has been documented and characterized, and the organisms, the C. elegans themselves, are transparent throughout the life cycle, which where cells undergo a very specific set of cell division at each point to produce the adult body. So genetic mutations of various genes can be monitored and examined their response, how that changes the development of the organism can be examined very easily all throughout its life cycle. Another one that's probably studied the longest uh, amongst the higher organisms is the Drosophila melanogaster, which is a fruit fly. It's been studied in a lot of detail because it only has 4 chromosomes. They have all been very well characterized. They've been sequenced for a long time, and it is very easy to manipulate them. So it has allowed them to study it to examine development as well as physiology and even behavior. You can create drunk flies, you can create depressed flies. So because they can be manipulated so easily and the chromosomes are so few, it is very easy to work with them. You also have more model organisms that more closely relate to humans, like mice, that are used very often in, again, genetic research because you know, they are again closely related to humans. However, there are some obviously difficulties when we're working with mice. They have a little bit slower life cycle, so you have to grow them a bit. They are expensive because of that. And then there are obviously some ethical considerations to think about as well. Another this is a relatively new model system that people have come up with, is the zebrafish, Daniel Rio. And this is great again for examining development in a vertebrate organism, as opposed to C. elegans, which didn't have that. Unlike mice, zebrafish grow quicker. They have a lot of progeny at a time, which is great. And the embryos develop relatively quickly and entirely outside the mom in a transparent manner, which means that you can easily study the development of the organism. So it kind of mimics the same kind of positive notes from C. elegans in this development part. And then for plants, the biggest model system people use is Arabidopsis, which is a small weed, and it's widely studied both for development and genetic purposes. And that knowledge that we gain from that is then applied to our common crops and other plants that we use all the time, like wheat, rice, and corn. So, finishing up, we want to know why we choose a particular model system. The biggest considerations that we have are usually based on either expense, space, or supply needs. Typically, we want to use something that's small in size so we can have more of it in our labs. We want to choose something that has short life cycles so our experiments finish in our lifetime, finish quickly. And then we always want to choose something that can create lots of progeny, obviously, because the more offsprings you have, the more replicates you have, the better your results are going to be. If you can find something that is easily manipulated, easily genetically altered or changed or cloned, that's a big plus. Small genomes, small C value, like we talked about, is again going to be a big plus because it allows us to know more about that genome and to manipulate it easier. And finally, if you are working on a human condition, you want that your organism can replicate some of those, is somehow relatable to that human phenotype or the human form that we're going to be studying at the end of the day. So, a summary from this week's lecture. Basically, we want to remember that Mendel demonstrated that heredity involved discrete heritable factors that affected specific traits, and genes were that basic unit of inheritance. The transfer of genetic information from bacteria and viruses into bacteria demonstrated this fact that DNA is the genetic material, and that later on other experiments told us how genes were the ones that were leading to the traits. DNA is a double helix made of two antiparallel strands of bases on a sugar phosphate backbone. And the two bases on those sugar phosphate backbones interact with each other through hydrogen bonding, ensuring that you have complementarity of strands that are then replicated through semi-conservative replication. Not all DNA in an organism contains genes, so you have a lot of extraneous stuff. It's not extra. It has functions, some of which are known and some are yet unknown. A lot of it is used for regulation and for control of gene expression. And then finally, you have several model organisms that help us learn about genetics in basic and applied research in biology, agriculture, and medicine, and that are essential to our understanding and progression of genetic knowledge. So next week, we are going to continue on this journey by working on the next section, which is going to be still going in the same book. We are going to be looking at chromosomes and mitosis and meiosis. So we are going to focus more inward, and we're going to specifically start looking at chromosomes and how they replicate and divide. So until next week, have a good week, and I'll talk with you then.